now we can move on to the second lobster, which is all about genetic transfer in bacteria. And so in the simulation, you're actually playing a game off and on throughout the entire simulation. And you are representing the world population, and your opponent is playing the pathogen. And your goal is to prevent the entire population from being wiped out by an infection. And as you take turns, you get to learn about different aspects of antibiotic resistance and the different aspects. In order to play the game, you keep learning more and more, and then as that happens, you will learn about the different types of genetic transfer. So the other scenario in the lobster simulation is that you will perform several experiments to help figure out what's going on with this patient. A patient is sick with an infection, and the antibiotics don't seem to be working on the infection. And so they give you his catheter, and you look at the patient's catheter sample, and you actually find all sorts of stuff in that sample. And so you need to perform different experiments to deal with the different components. And so his infection is with a Bomani, and it turns out that he also has some resident Pseudomonas aeruginosa, but the infection that you're trying to kill is actually the A. Bomani. And also in this sample is a bacteriophage and some naked DNA and some waste. Okay, so make sure that for each of these experiments, you can tell me what are you testing for, how did you set up the experiment, and then what results did you obtain, and how did you interpret those results. Okay. All right, so now I'm going to go over the 3 types of genetic transfer. Okay, so at the end of this lesson, you should be able to differentiate between horizontal and vertical gene transfer, and you should be able to compare the 3 main mechanisms of horizontal gene transfer in bacteria, meaning you should know all the steps of each type of horizontal gene transfer that I'm going to go over. And then you should be able to describe the role of plasmids in genetic recombination. Okay, so horizontal gene transfer is the movement of genetic material from one organism to the next. This is very different from just parent to offspring, which would be vertical, passing from one generation to the next. So horizontal gene transfers all the peers, all the bacteria that are already alive, okay? That's why it's horizontal. So this can be between members of the same species or not. It doesn't even have to be the same species in order for horizontal gene transfer to occur. And we say closely related species, but it appears that I mean, where do we draw the line? How closely do they even need to be related? That I don't know if we have an answer to that quite yet. Okay, so contrast to vertical transfer, as I already mentioned, which would be from parent to offspring, so down. Horizontal gene transfer, on the other hand, is side to side, horizontal peers. Okay, and there are 3 main mechanisms that I'm going to talk about for horizontal gene transfer, and those are conjugation. And the first thing you should think of, or the first 2 things you should think of when you hear conjugation, you should think sex pilus and you should think plasmid. Okay. Second mechanism of horizontal gene transfer that I'm going to talk about is transformation. When you hear transformation, really you should think of the experiment you learned in lecture called Griffith's transformation with mice, but if you didn't think about that, that's okay, as long as you think of naked DNA, taking up naked DNA. And then number 3 is transduction. And when you see transduction, you should think of that bacteriophage. And so one thing that you can do when you're studying is next to the word conjugation, you could draw a plasmid. You could even draw 2 bacteria with a pilus attaching them in between. Okay. When you have transformation, you could put little pieces of DNA outside of a cell. And transduction, you can draw a little bacteriophage. Okay, so ready? We're going to go over the steps. So conjugation, and I think this is all review. I think you already talked about this in lecture. Okay, so in conjugation, you have one cell that has a plasmid with the genes. In order for conjugation to occur, the genes are called the F factor. So this the plasmid can be referred to as the F factor plasmid, but it can also have other genes on it as well, like resistance genes, such as antibiotic resistance. Plasmids, as you recall, are completely separate from your chromosome. The chromosome is all the day-to-day normal metabolism, what we call housekeeping, just what the basic requirement for life for that cell. Plasmids are special extra stuff, right? And if you have the F factor, you're referred to as an F plus cell. The F plus cell will extend the pilus, and that pilus will touch the F minus cell and grab it. And then a mating bridge will form, which is just more secure, holds them tight together. And then that mating bridge is where a copy of the F factor will get transferred through into the F minus cell. Okay, so the first step is to make cell-to-cell contact. That's the F plus cell has to extend the pilus. So it's going to adhere to the neighboring cell, the F minus cell, via the pilus, and that's going to attach and pull it a little bit closer. And then you form the mating bridge, right? So that's everything I already said. So here's an image. So this would be the beginning, right? They're pretty far apart. So this F plus cell has just extended its sex pilus and adhered to the F minus cell, and now it's going to pull it closer, and then it's going to form that mating bridge. So here's the mating bridge. See how much closer they are together, and this is also a thicker in diameter. And here it even looks super thick, right? It looks almost like this is a cell that's dividing, but it's actually a mating bridge between 2 bacteria. Okay, so continuing on with our process of conjugation, once that mating bridge is established, you're going to have simultaneous transfer and replication of the plasmid. The F plus cell doesn't want to lose its plasmid. It's going to make a copy. And so it's actually a plasmid, just like the chromosome is double-stranded. So you actually, a little nick will happen in the plasmid, and one strand will start to be transferred. And as that strand is being transferred, a new strand is going to be synthesized, so that when conjugation's finished, your original cell still has double-stranded plasmid. And then as the recipient receives the copy of the plasmid, it will also immediately start replicating so that it will have a double-stranded plasmid at the end as well. Okay, so simultaneous transfer and replication, one strand of the plasmid, the donor, is nicked and transferred through the bridge, simultaneously copying and replacing that copy. And as well, the recipient will also copy its plasmid, so it has double-stranded plasmid. So both of these will have double-stranded plasmid. Okay. And of course, the newly acquired plasmid will make this now also an F+ cell. And the transfer is complete. They both have double-stranded. I know it's only shown as one here. You could draw on this. You could modify these images to show the 2 strands if you want. Or you could just make a note, make lots of notes so you don't forget that it is actually double-stranded. Transfer is complete. You have 2 F+ cells. Whatever genes were on this plasmid, now both of these cells have. When these cells divide, like when they produce offspring, when they undergo binary fission and divide, they will copy both the chromosome and the plasmid. So then this plasmid will get passed down from now on. It will get passed down through vertical gene transfer, but also each of these cells can continue to form, to continue to perform conjugation and spread the plasmid to other bacteria. So you can see how fast, how rapidly bacteria in a population can be changed to have special traits through the process of conjugation. So we believe that conjugation is the primary method in which antibiotic resistance is actually formed and passed on among bacteria. Okay, so both cells at the end will have double-stranded F factor plasmid and whatever other special genes are encoded in that plasmid. Now, a cell can have more than one plasmid. It can have several plasmids, and as long as it has the F+ plasmid, it can form this bridge, and it can share all of those plasmids in the same manner, right? And then once conjugation's done, the bridge will disassemble, and these can both perform conjugation with other F- cells. Okay, if you want to see the process that I just went over of conjugation, here is a link. I think when you pull up this link, it's going to be a list of videos, and then you would have to select the conjugation video. This also, it might be too old now, and you might not have that, but I also have this animation. So if this link doesn't work, don't sweat it. You can use this one. And in fact, we'll go ahead and watch that right now before I forget. So this should be real brief, just 2 minutes. Excuse the mandatory edit. Okay.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of 2 different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
All right. So that was just as I had said. Okay, so next we're going to talk about bacterial transformation. Okay, so I already said this in the video, I already said this, but the F factor are the genes that are needed for conjugation to be able to occur. So being able to make that mating bridge, being able to make the pilus, copying the plasmid, everything that's needed is called the F factor, and it can contain other genes, or there can be additional plasmids that have, if it has any type of antibiotic resistance, it'd be an R factor. If it had heat resistance, it would also be a type of resistance, so that would also be R factor. And usually we do find R factor plasmids that include the F factor gene, so we see it combined into one plasmid, but it's also not uncommon to have more than one plasmid. Okay, so the second type of mechanism for horizontal gene transfer is called transformation. This is when a cell takes up DNA that's just in its environment, left over from like dead bacteria, and it takes it in and it recombines with its own chromosome. And so the steps of that would be first the cell needs to be able to bind the double-stranded DNA from its environment. It needs to be able to bind to the cell membrane, and that's going to lead to taking that inside of the cell. And but first, before it can take it inside of the cell, it needs to break it into small pieces. So you bind the double-stranded DNA to the cell membrane, you fragment the double-stranded DNA into smaller pieces, and then as you transport the smaller pieces in, you actually will take apart the double-stranded DNA to degrade one of the strands and only let one single strand in. So a single strand, small piece of DNA comes into the cytoplasm, and if it's allowed to go near the chromosome, and if it can find a homologous region, then recombination can occur, but it doesn't always occur. And so if it doesn't occur, what will happen instead is that it will chop up or hydrolyze that foreign single-stranded DNA, okay? And so this is the natural way that cells do this, but if you wanted to force this to happen in the laboratory, you can force cells to take up external DNA through some unnatural methods like electroporation, heat shock, or even calcium ions. But the natural way that cells do this, usually when they're starving and they need to have an alternative food source, they can actually use external DNA in the environment and chop it up into pieces and use it as a carbon source and as building blocks. And so sometimes when it's doing this as this starvation technique to survive, some of the single-stranded DNA ends up recombining with the chromosome instead of being used as food. And sometimes that ends up being beneficial. And if it's beneficial, then that trait will be passed on to the next generation. Okay, so what that looks like in a simplified version, so you have this fragment of DNA and it's going to bind, and it's not shown here, but it, you know, we saw the details on the previous slide, but here it just says it takes up the donor DNA, which is just out in the environment. Okay, and it takes it in, and you know that it would actually be single-stranded. So this is a little outdated, but it'd be single-stranded. And if there's a corresponding region called homologous region, then it can line up and it can actually swap out pieces of the foreign DNA with its native DNA. Another way that it can do this is actually the one end binds to the chromosome, and then the other end binds to the other region of the chromosome, and so it inserts the whole piece, and then it will be copied. So you can have recombination through kind of like crossing over, but you can also have it through like just actually incision of the whole little chunk. Okay, and look here, it's showing that it's just a few nucleotides long. So they are small pieces, and so this would have to happen enough before you gain an entire new trait, right? But again, this is just a simplified diagram, just to help you see kind of the general idea of what's going on. But you need to know the detail that I put here on this slide, okay, and here. Okay, so let's go ahead and watch this short animation on transformation. Another mandatory ad. Okay, there we go.
A pathogenic S strain of pneumococcus forms smooth colonies on Petri plates because the bacteria are surrounded by slippery polysaccharide capsules. A non-pathogenic R strain lacks the capsule and forms rough colonies. An R strain can be transformed into an S strain. In this procedure, DNA is extracted from the S strain. The purified S DNA is added to a culture of the R strain bacteria. The bacteria take up fragments of DNA from the environment. The DNA passes through the cell wall and plasma membrane. One strand of the foreign DNA degrades. Recombination between the foreign DNA strand and the bacterial chromosome is accomplished by a pair of crossing over events. The result is a double-stranded DNA in which the two strands are not complementary because part of one strand came from a different source. The leftover DNA is degraded. Both strands of the chromosome replicate, so the two new DNA molecules are genetically different. One daughter cell is genetically the same as the original cell, and the other contains a DNA code that results in the formation of a capsule around the bacterium, resulting in pathogenicity.
Okay, so the bacteria that gains the foreign DNA is going to become the pathogenic bacteria because it now has the gene for that special coat. Okay, so I know that went really fast. You can rewatch that video on your own because you have the link. Okay, last method is called transduction. Then I will be done with the introductions and I'll take your questions. Okay, so for transduction, oh yes, in this, before getting to transduction, I did include another slide to show that how that crossing over, what that looks like, which was also shown in that animation we just watched, so I won't go over this, but you can look over this. All right, so horizontal gene transfer mechanism number 3, transduction. This is using a virus called the bacteriophage. Okay, and it's going to involve 2 hosts, 2 bacterial cells. We'll call the first one the donor cell, and the second bacterial cell the recipient cell. So you have a phage, a bacteriophage, and it injects its phage DNA into the first bacterial cell, and it goes through the replication process. As it goes through the replication process, what happens is it accidentally takes up some of the bacterial DNA, and so you get some phage viruses that now have both bacterial and viral DNA. In this image, it's only showing one or the other, but it would actually mostly have the virus genome, but it also has some bacterial genome in it. And so then when it goes on to its next host, it's going to inject that DNA, which because it came from a bacteria already, is going to have some similar regions. So it's going to be easy for recombination to occur from this process. And so what's shown here is the recombination event. So you have, you now have foreign DNA from that first host has been added into the bacterial chromosome. And then when that bacteria undergoes binary fission, as it always does, it will copy that whole chromosome. And so now you'll have 2 identical bacteria cells, both with the donor bacterial DNA and the original DNA. And that will continue on and on as it divides. Okay, so this is using a phage that accidentally takes host DNA with it to its next host, and then you get a recombination event and it becomes part of the new host's chromosome, and then that will be spread to offspring through regular binary fission. Okay, and so let's go ahead and watch that last animation.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Okay. So let's go ahead and close this.
Grammarly does more.
Okay. All right. So the next slide I included is just a quick little comparison of the 3 types of horizontal gene transfer so that you can have a nice quick visual. Transformation, taking up naked DNA from the environment. Transduction uses a bacteriophage to take genetic material from one host to the next. And then conjugation is between 2 bacterial cells via pilus to share plasmids. Okay. All right. So the topic of antibiotic resistance is that's the main topic in looking at each of these horizontal gene transfer mechanisms to see if they were responsible for antibiotic resistance. And so I just wanted to give you a quick little snippet about antibiotic resistance. I know we talked about it last week. It was in your simulations last week, and you've talked about this in lecture, but I just want to remind you that antibiotic resistance is a very serious issue. We already have almost 3 million people in the US alone die every year from antibiotic resistant infections, and this is growing rapidly. So when I taught this last summer, not this summer, but previous summer, so a year and some months ago, this number was approaching 2 million. So in just 1 year, it's already 2.8 million. So this is very rapidly occurring, okay? From the 2.8 million infections that are resistant, we've had at least 35,000 deaths. And again, this is only in the United States. So we are seeing more and more strains of bacteria that are resistant to not just one, but multiple categories of antibiotics. And then we call that MDR, multi-drug resistant. And so this is becoming more and more of an issue. And the majority of people that contract a multi-drug resistant infection get these from hospitals or healthcare settings like nursing homes, for example. So when you guys become healthcare professionals, please take very, very great care to prevent infection, first of all, right? Making sure you keep all surgical wounds and any other wounds very clean. You check for those infections, catch your infections early on, and always take a culture sample and make sure that the tests are run to find out which antibiotics will treat the infection. And also make sure that patients understand to use the full dose of the antibiotic, and again, making sure it's the correct antibiotic, and that the infection is clearing up, right? So prevention is most key, and good practices to prevent infection will save lives. So that's should be, if you get nothing else out of this class, it should be that, right? Save your patients' lives by having really, really excellent practices. You will be taught the correct procedures for everything, and it'll be up to you to follow them to a T, okay? All right, DNA technology. Knowing how bacteria do things, we can then take those mechanisms and control them, manipulate them, so that bacteria and other organisms can produce stuff for us. So recombinant DNA technology is taking one or more genes of interest, inserting them into an organism, and having that organism produce the protein for us. And that's another way to say that is genetic engineering. And that's what you're doing in the simulation to produce the erythropoietin. So inserting the desired genes into the host chromosome. We do this all the time with insulin. Back in the day, when we first learned about insulin, we used to get it from cows, and chickens, and pigs. But some people would be okay, but a lot of people, their immune system would reject the insulin. It would say it's foreign. They would attack it. So it wasn't until we were able to do this DNA technology and get the human form of insulin growing, and we can now grow them in like rabbits and things that are easier to work with. And so we can have safe insulin for people. This is also, this recombinant DNA technology is also how we make our hepatitis B vaccines. And of course, there's more applications and uses, but those are the two most important. Okay, so when you do the simulation, towards the end, there's going to be a quiz question that says something about like which of the following is not required for transduction. I think that's how it worded it. And it actually gave 2 correct options, like it gives you 4 options and 2 of them were correct, but it will only accept 1 of them as being correct. So that was, maybe that won't happen for you when you do it, but that is what I encountered. And so I wanted to make sure to give you a heads up. So if you see this question and it gives you the option of competent cells and donor bacteria and recipient bacteria to be in close proximity, the one that it's looking for is this. So in transduction, the donor and recipient bacteria cells don't have to ever come in contact because the virus, the bacteriophage, is going to come in contact with each of them. Okay, and actually, you don't need competent cells for transduction, right? You only need competent cells for transformation. So this is also a correct answer in that it's not required for transduction. But the simulation, it was specifically looking for the second one. So I don't know, they made a mistake or random mistake appeared in mine, because when they generate these, there's like some variation between each player to try to mix answers up and things like that. So I don't know, maybe you won't have this problem, but I wanted to let you know just in case.